Okay, so I've started the recording. So if you remember, so let's quickly have a recap of what we do. I see Shagufta has also joined so that she can also get to know what exactly we discussed last week.
Yeah, hi.
So couple of things that we discussed is before we jump onto the clinical SAS concepts, it's better to understand what exactly is clinical data management, uh, who are the people involved, how is the entire clinical trial, how does the entire clinical trial happens, because this clinical science part comes after the clinical trial is um, performed, basically on the data of the on the patient and the lab data. So I'll share my screen quickly. So let me know if you are able to see my screen.
Yeah. Yes, I can.
Okay. So clinical data management. So first comes the clinical data management, where exactly the trial is taking place. When the clinical trial takes place, the patient comes to the hospitals, or when the patient comes to the hospital, the patient provides their data, and the nurses take the data, process it, and once all the patient data which has been targeted for that clinical trial has been accomplished, or the data is locked, the entire data is locked, and we go ahead with clinical statistical program. Okay, we extract the data from the database of the application, of the we extract the database, put it in clinical SAS, put it in SAS system, and then we start creating the reports, the listings, and the graphs. So this is the entire thing. Now, how does a clinical trial happens? It suddenly doesn't happen, right? So there are certain things which are there. So suppose for a medical device company, or suppose for a pharmaceutical company, there is an R&D section who's constantly researching about new drugs, what new drugs can be, what are the different therapeutic areas where these drugs can be beneficial to, and they are constantly researching. So the R&D or the R&D section of any pharmaceutical companies. By pharmaceutical companies, I mean Novartis, AstraZeneca, Merck, Novo Nordisk, all these things. Okay, all these are companies because they are mainly the medicine companies that we have, right? The medicine that we take, if we look at the back of the medicines, these are the companies which generally create that. So the R&D division creates a medicine or creates a formulation. That formulation is first tested on animals. After that, it is tested on animals, the safety is measured, the minimum dosage is measured, and what is the outcome of the dose on the animals is measured, and then this report is submitted to the Food and Drug Administration, which is the FDA. Okay, this is called the preclinical trial. Once FDA looks at the reports and gives a go ahead that okay, go ahead. I mean, you can go ahead with the clinical trials on humans, then this entire clinical trial on humans starts to take place. Now, within this clinical trial entire concept, there are three segments. One is the sponsors. Who are the sponsors? These are the pharmaceutical companies like Novartis, Merck, J&J, AstraZeneca. Institution and organizations that initiate, monitor, and finance the trial, because this clinical trial, conducting this clinical trial is very costly. Sponsors are the big shot pharmaceutical companies who sponsor the clinical trials. Okay, that's number one. Second is CRO. What are CRO? Contract research organization. What are contract research organizations? These are organizations whom the pharmaceutical companies reach out to or have a relationship from previous. I mean, will already have a relationship uh, from the beginning, and they outsource the clinical trial to them. Outsource means what? So I'll give you an example. So we all go to office. So we have a specific office, X, Y, Z, whatever. After office, you see there are cab systems which are available that we can. I mean, there are. I mean, the person can board a cab, and the company, uh, or yeah, the company helps that person to reach home using the cab. So there are a lot of cabs which are there because after 7 o'clock or after 8 o'clock at the night, there are a lot of people who return home, and organization provides the cab services. You will see that generally or the organization for whom the people are working does not own the cab services. They outsource it to different vendors. That there is a separate vendor XYZ, there's a separate vendor ABC. They have a lot of cabs in place. Okay, so they come to a pact with the sponsor or with the organization and help them to provide cabs for the pickup and drop of the employees. It's exactly the same thing. Contract research organizations are research entity hired by sponsor to plan and run this entire investigation or clinical trial. Like they say that, okay, this is what we are trying to create. I mean, this is some, I mean, we are trying to conduct a clinical trial on this particular drug. We will be needing your help to conduct this clinical trial in two places in Europe, two places in US, two places in India, three places in Japan, something like that. You help us do that. You coordinate it. You have a system where you can capture the data. You send investigators, you send data monitors, you send people to the sites, you select the patients, you take the data, and you complete the entire clinical trial. Okay? And next comes the sites. Sites are nothing but basically the hospitals where the clinical trial is basically taking place or generally takes place. Centers coordinating data collection from trial patients. So if I have to give my trial data, where do I do? I have to go to a hospital and give it. I have to go to otherwise it's difficult. So the hospitals are basically the sites. So once you take the medicine, suppose first you go to a hospital, suppose Apollo hospitals or suppose Manipal hospitals, you give your data. Like this is my demographic data, this is my medical history data. That is the first day that you go. The day when you go to the hospital, it's termed as a visit. You are visiting the hospital or you are making a visit at the hospital. Again, you make a second visit after 3 days. That is where you take the drug. Okay. And before taking the drug on the second visit, you give your informed consent that, okay, I am a person who is willingly wanting to take part in this clinical trial after going through all the rules and regulations of the clinical trial. That is the most important thing. Informed consent, it's called. I give the informed consent. I go through an inclusion exclusion criteria. What is inclusion exclusion? All the criteria are there are set of questions which are there. I'll show you about it. There are questions which are there. Like, are you pregnant? Are you lactating? Or do you have cancer? Or are there any physical disability? If you have all these things, you will not be eligible to take part in the trial. And if you don't have anything, also, I mean, if you don't have problems or these criteria, then you are eligible to participate. So on one visit, I go, give my informed consent, answer all these questions, and if I am eligible, I come back. Again, I go back on a third visit and take the drug. So every time a patient visits a hospital, it's termed as a visit. So this is basically an entire scenario of a clinical trial that takes place. Got it? Any questions?
No.
Okay. Okay, I'll go ahead. So this is the entire flowchart of clinical trial, and we will be discussing this at the last. So statistical analysis is a part which happens after we go and run, and then comes the statistical analysis part, where exactly we talk about clinical SAS. Okay. So if you see the study initiation, so again, R&D, there is an R&D department within a pharmaceutical company. They have created a formulation and they have tested on animals. After they have tested on animals, where do they go? After they tested on animals, they send the report to FDA. After they send the report to FDA, the FDA looks into the report. FDA gives a go ahead that okay, fine, this is correct. You can start a clinical trial on humans. When you are starting a clinical trial on humans, the first activity which happens within the first activity which happens as a part of the clinical trial is the study initiation, where a protocol is developed and approved. Now, what is a protocol? Let's quickly check that. Protocol is nothing but this is basically a protocol. If you see, okay, this is basically a protocol where you have all the data related to everything, like entire trial. Okay, if you see, you have everything related to the entire trial. If you see, so schedule of activities, study rationale, background, non-clinical investigations, clinical investigations, risk benefits, what are the points. So this is basically the Bible for a study. Like everything will be written there. If you go down, what are the doses? What is the summary of it? Then afterwards, if you go down more, how will the study treatment and schedule will be done? What are the how will be the treatment groups be divided? Okay. Then if you go more down, what is the study rationale? Like why is the study being conducted? What will be helpful from this study? Where will be the study will be conducted? What are the what are the drugs which will be involved in the study? What will be the background? Everything. What is the investigation? What is the clinical experience with a vaccine? If it's a vaccine trial, so we will have basically all the literature based on, or all the data within the protocol based on the vaccines. Okay, so on and so forth. So you have this as a protocol. So this is something where the sponsors or the pharmaceutical organizations write about it. Write about it means the pharmaceutical organizations devise this protocol. They have a team of very highly qualified individuals who come together, sit, discuss about the study, discuss about the drug, and then create this protocol. Okay, this is the first thing. And once this protocol is developed, this is again sent to the FDA before the start of the clinical trial so that it is approved. Now, once the protocol is approved, it is a go ahead that you can start and go ahead with the clinical trial. Okay, so once a go ahead is given, then the study planning starts. What is the study plan? Study plan is the site is selected. Just give me a minute. Correct. So site is selected. So you have to select, right? Where the, I mean, you have to select the hospitals and the sites where you want to conduct your clinical trial, where the patients will come in. Now, suppose I give you an example. Suppose there is a cancer study which is being conducted. A study is being conducted to test oncology drug on cancer patients. So, don't you think that the study should be conducted in sites or hospitals which are equipped in handling cancer patients, and which basically has a large number of cancer patient footfalls? Don't you think so? So the sites should be selected on that basis. Suppose you have an oncology trial or cancer related trial, and you are selecting a hospital where the hospital is basically famous for vaccines. So that won't be beneficial for the study, right? Or that will be beneficial for the sponsor as well. So these are some of the investigations, or these are some of the ideas or selections that you have to make when you are planning the study. You have to look into which therapeutic study are we doing it. By therapeutic study, I mean, is it a cancer study? Is it a cardiovascular study? Is it a nervous system study? Is it an oncology study? If this is a study, if the study is going on these therapeutic areas, then we or the CROs, because it's the sponsor has outsourced the clinical trial to CRO, it's the CRO's work along with the sponsors also to jointly find out the sites where there would be patients, where there would be large number of patients who would be coming in, or who actually reside close to that site, having that particular therapeutic area issue, or having issues related to that particular therapeutic area. So if you are doing an oncology study and you select a site, and close to that site all the people are all the people who reside close to that site have diabetes, then there's no point, right, doing it. For oncology study, it's better you do it at Tata Cancer Hospital, where there is more cancer patients visiting every day, various types of cancer patients, breast cancer, lung cancer, all those things, for patients visiting every day, so that it would be easier for you to conduct the trial and to easier for you to get the patients. Got it? Any questions here?
No.
Okay, fine. Next is the patient recruitment. You have to select the patients. Who are the patients that you would likely to consider in a clinical trial? Like all patients who ever have cancer, you can't take them, right? So there is some criteria which is there, based on which you will be selecting those. Okay, so that is the thing. And what is the patient? There is patient recruitment and investigator selection. What is investigator selection? Investigator selection is a concept where investigators are the people who are responsible to actually conduct the clinical trial. Not conduct, I would say, actually oversee the clinical trial from a leadership angle. So there is a project manager, right, happening in every project. They are not actively doing hands-on work, but they are looking into the deadlines, looking into the timelines. If there are any issues with any of the fellow colleagues, they help them out with completing the project, with completing the task. Investigator is something like that. So there is a principal investigator who actually leads the overall clinical trial. There is a lead investigator who actually oversees the clinical trial from site specific. Suppose for every site there is a lead investigator, and for 2-3 sites there would be one principal investigator who would be responsible for overseeing the entire clinical trial conduct phase from a leadership level for 2-3 sites. And the lead investigator is a person who would be basically generally looking into those site specific. So for a site, for Apollo site, there is a lead investigator who is looking into that, looking into all the operational work, like whether every patient is coming, whether every patient is treated correctly, whether every patient, the data is taken correctly, that whether every patient has been signing the e-consent, everything is done. Okay, this is one. Then come the study. So one, everything is selected, the patients are selected, the sites are selected, the investigation selected. Then comes to study conduct phase. Study conduct is what? This is where the CRF is designed. CRF is case report forms. Okay, and this is where you will come across an EDC system, which is called an electronic data capture system. Okay, so previously what happened was that there were questions. So what are CRF? Case report forms are nothing but set of questionnaires which are given to the patients or asked to the patients during the course of the clinical trial, and that data is stored in a system, and later on, once the clinical trial is completed, that those answer to those questions are extracted, and based on those answers, analysis is done, where we would be doing, which we would be actually doing as a part of this course. Okay. So if I show you a case report form quickly. So see, so this is an example of a case report form. So see, so it is asked for every, this is a list of questions. So this is a demographic question. Okay. Okay, this is a demographic question which is there. So what is the visit date? What is the sex of the participant? What is the date of birth? What is the current age? What is the ethnicity? All these questions are asked to every participant who comes in. After that, these are the informed consent. Okay. Did the volunteer provide a signed written consent? There should be a signed written consent which is there. And what was the time of the consent? Because if you do not have informed consent, if you see that if any one of them is no, inclusion alert if no. So if there is any of these is no, then the patient will not qualify to participate. You will have to have provide consent form. Was the understanding checklist completed with 10 questions answered correctly answered? Was consent form for female participant of childbearing potential completed? So the female who are participating, they have to fill up a separate childbearing potential form. There is a separate 10 questions form which needs to be filled up by both male and female. So all these things has to be done. If and only if they are completed, then only the person will be participating. So previously what happened, I'll give you an example. Suppose I am a patient. I used to come in and I used to give these data. Like this is today's the visit date, my sex is male, my date of birth is this. And these were captured in this paper, like in this PDF only. Or the printout of the PDF was taken and they were, they were written by hand and kept it. So suppose there are 20 CRFs which are there, and for only one patient, 20 CRFs had to be filled up. So for a phase 2 trial, there are at least around 100 to 200 patients. So imagine the amount of paperwork. So that was actually an extremely delayed process and an inefficient and ineffective process. From there, it shifted to an electronic data capture system. You all can do some Google search, what exactly is an EDC system, how it works, what are the EDC systems which are in place. So this is one info, you all can Google search that and you'll get to know what exactly is EDC systems are and what are the types of EDC systems are there in the market. Okay, so this is one part. Next part, what we go here is, once we have this, so this is a CRF which is there on Word document, which you can see. All these questions are transcribed in a digital format in eCRF. Okay, all these transcribed means an EDC system is open. There is a person sitting in front of the EDC system, an application, and then one after the other, all these questions are configured within that EDC system, so that it's a one-time configuration. Whenever a patient comes in, all those questions would be filled up and submitted. Then another form will be opened up for a fresh participant. So everything is saved electronically and digitally. So the quality of the data would be good, the retention of the data would be long, and also it is efficient and effective. And once this study is configured based on these eCRFs and all these questions and everything, data capture starts happening. Data capture starts happening means what? At the sites, nurses are there, investigators are there, patients start coming in, and the data is starting to come. Like the patients are asked those questions, the data is taken, and the data is submitted in the EDC system. The study starts. Basically, the study goes live. Clear up till now? So if you see here, what is CRF design? Case report forms, they are paper CRFs, and then when they are transcribed into a digital format, they are called eCRFs. Capture data in database tables through EDC systems. So all the data which are electronic database, whenever they are, electronic CRFs, whenever they are configured within EDC systems, and the data starts coming in, those data are basically stored at the backend in tables and table format. And those are basically called Rave, Oracle, Clinical, Inform. So EDC system example are these: Rave, Oracle, Clinical, Inform, Viva, Vault, and so on and so forth. Okay. What is database design? Database design are these edit checks. We have these edit checks which are there: edit checks, derivation, and conditional rules. Now, the moment I say that, how do I explain it quickly? I'll explain it. If you see the current age, if someone provides the current age less than, so see, if it's auto-calculated means what? So see, the sophistication of the clinical trials that have happened over the years of capturing the data. So if you give the date of birth here, okay, so not in this Word document. So you have transcribed these fields in the EDC system, and you have written a logic that the moment you give this date of birth, the logic will subtract the date of birth from today's date and provide you the current age. And if the current age is less than 18 and greater than 45, it will give you a message that does not qualify to participate, because this study only allows people within the age limit 18 to 45 to participate in this study. So this is called an edit check. And this is called a auto derivation. Like if you give the date of birth, automatically the age will be derived. So chances of error is less. Okay. So if you see sex is female, if you take, see, automatically the birth control CRF becomes available. Previously, these would not happen. So if you are a female, the birth control comes because you will automatically understand, okay, this is a female, so she has to populate the birth control CRF. Okay. Now, if we go to the birth control CRF, what exactly is birth control? Let's see what exactly it is. Is the subject is the subject child potential is the subject of childbearing potential? Yes or no? If you give no, it's not childbearing. The following field becomes available. What is the reason that the person cannot have childbearing potential? Tubal ligation, bilateral oophorectomy, hyperectomy, postmenopause for 1 year. And if it is yes, she has a childbearing potential, immediately the following phase, when was the last menstrual period, this date comes in. So everything is not given at, everything is not visible at one go. One after the other, as you progress or as you fill up the questions, the second question comes in. So there is very limited chance of entering wrong data. Because previously what used to happen is lot of bad junk data used to be filled up. And as a result, the data monitor, the person from the sponsor's end who used to monitor the trial, he or she had to do a lot of work in segregating the data, looking into the data. If there is wrong data, send the data again to the site to correct them. All these things took a lot of time. It was a lot of expense and everything. But with this digitization and with all these sophisticated EDC tools coming in place, the the data error has drastically reduced a lot. Got it? Once this is done, data is captured and everything, which I was saying, the data monitor, still we need data monitor. So data monitor would do. So data monitors what? Validate data entered by the data entry. So there is a validation which would be done. Like, is all, I mean, more or less, are the data correct which has been entered? If there are any data which is not correct, they has to be corrected. That is called a query. The data monitor raises a query. Okay. After the data monitor raises a query, that is rectified. After that is rectified, just a minute. Yeah. So after this is rectified, all the query, so the data comes in a good shape. Also, there is one more thing, is adverse event tracking. So during the course of the entire clinical trial, there would be people who would be taking the drug. There will be adverse events which would be reported because there would be some headaches of some patients happening. There would be erythema which is happening. There would be some blood clots which would be happening. There would be some vomiting which would be happening. There would be some pain which would be happening. So on and so forth, right? So these are basically the adverse event tracking which happens. And all those adverse events are medically coded to a medical dictionary. Okay, because the adverse events which are reported will come in different forms, or I mean different languages. Like those are not standardized. They has to be standardized with the help of a medical dictionary called Medra. Medra dictionary. MEDDR. Take a look at that. Okay, this is the one. And once all these data is verified and looked into, and all the last patient has come and given the data and everything is done, the study is locked. Study is closed. So after that, you cannot make any changes to the study because all the data that has been collected as a part of the clinical trial will be extracted, and on that data, the clinical statistical reports for safety and efficacy will be created and sent to the FDA. Clear? So this is the closeout phase where you lock the database and then you perform the statistical analysis, and then the based on that reports, safety and efficacy reports, there is a medical writing team who is there, who will write the synopsis of the study reports, overall summary, side effects, and submit it to the FDA or drug control or whoever is the approving body. Any questions?
Um, Ajit, I have one question.
Go ahead.
I just wanted to ask, like this study and this, like our role is the statistical analysis. So it will go with each other, like they run each together, or like after.
After
The,
data, yes. Actually, what happens is that you have a very valid questions. Now, what's being done is as and when the data starts coming in. Okay. So previously, what used to happen is once the study was locked, database was locked. After that, once the database is locked, after that, the statistical analysis used to create the specification. Like specifications were created from the, specifications were started being, formula, started being designed from like during the study, during the clinical trials conduct phase only. But right now, what's happened, but like, one second. Specifications were written during the course of the study conduct. This is what I'm talking about, what used to happen previously. And the analysis was performed only after the study. But right now, what happens is that during the course of the clinical trial as well, there are some interim reports which are created to see what is the efficacy, is the safety correct or not, to understand where exactly the study is lying, or is the study going in the right direction or not, based on
yeah.
the reports, based on the safety reports. Those are the interim reports which are created during the course of the study. But a final report is obviously created once and after the entire uh, clinical study is locked or clinical trial is locked. That is where a final analysis, like during the course of the study, the SAS programs are written, they are validated against the reports, validated against the data, so on and so forth happens. Interim reports are created so that the sponsors know that, okay, these are the safety reports, these are the efficacy reports which we can expect, these are the number of people who has been given concomitant medications as a course of the adverse events. These are generally the medical history of the participants who have come in. So these are some of the endpoints, or not endpoints, I would say. These are some of the checkpoints which the sponsors would like to see during the course of the clinical trials. So this now run in parallel, the clinical data management part and the clinical statistical part. Okay. But the final report creation and the analysis and the full package of SDTM, ADAM, table listing figures and everything is submitted to the FDA and also to the sponsors after the study, after the database lock happens. Like the study clinical trial data is all in the EDC system and the database is locked, and the clinical trial is like the clinical data management part of the clinical trial is locked or completed.
Okay. So you mean to say like till phase three, after phase three, the database lock will be there, and then it will submit to the FDA for the approval, and then phase four will be happen, right?
It's not phase three specifically. So if it's a phase one, you have to, for phase one also you have to share the results with everybody. There also will be a database log. If you are going with a for a phase two study,
Okay. So it,
for every phases the patients and the conditions changes. But for every phase, once the database is locked, you have to then do the analysis and submit the report to the FDA or whoever is the regulatory authority.
okay.
Okay.
Yeah. Yeah.
This is basically the roadmap, the research protocol roadmap which is there. So basically, what exactly the protocol has, what are the objectives of the clinical trial, what I mean, what are they planning to establish or planning to find out. as a part of the clinical trial which is being conducted. What are the objectives basically? What are the background and rationale? What is the subject selection criteria? What are the treatment plan? How will you be treating the participants? What is the response evaluation criteria? How will you evaluate the responses? How will you conduct the efficacy, so on and so forth? And there is a statistical section. But there is a statistical section is there in the protocol, but a separate how the statistical analysis will be done. All these are detailed in a separate document which is called the SAP, Statistical Analysis Plan. Okay. So these are some of the important pointers that we have. What is an informed consent if you see? Learning the key facts about a trial before deciding whether to participate. This is entirely for the participants. So research, study purpose, risk and benefits, alternative treatments, confidentiality of reports, all these things are mentioned as a part of the informed consent, and the participants are made to understand those points by the investigators or the clinical, I mean, or the leads who are present as a part of that clinical trial. Maybe the lead investigator or the principal investigator. Medical treatment available if injury occurs. So what if you are going in for a clinical trial, the participant before signing the informed consent form should be detailed about all these aspects. What are the research purpose of the study, risk and benefits, alternative treatments, whom to contact to answer the questions. Once the participant is confident and satisfied with the explanation and has gone through the consent form, then only he or she signs the form, and that is when that person becomes eligible to take part in the study. Not before that. Got it? This is a question, this is a pertinent question that can be asked. Okay. Like when does a participant becomes eligible? So this informed consent is one of the criteria. Got it? I'll go next. Serious adverse events. Yeah. So as a part of serious adverse events, what happens? So during the course of the trial, you take the medicine because this is a trial and test which is happening, right? So what happens is that events that occurred in any of the following. So death or life threatening. If a death happens, so that's the serious adverse events are there. Then there are serious adverse events. So serious adverse events happen under what conditions? If there is a death or a life threatening, if there is a hospitalization or prolonged hospitalization, if there is a persistence of significant disability which takes place, or if there is a congenital anomaly or birth defect which happens. Okay, then it results in serious adverse events. Events that are serious, unexpected, and related or possibly related to the participation in research must be reported to the sponsor, FDA, and IRB in a timely manner. So any kind of serious adverse event, suppose related to the drug or not. Suppose you are taking a drug and you meet with an accident. So that is something which that adverse event is not something which is related to the drug, right? But if you take the drug and if you have a heart attack, that is something might be related to the drug. Might. If you and you are hospitalized, if you take the drug and you see that your liver gets damaged and you have to have and it's life threatening and you have to be hospitalized. Those are some of the serious adverse events which has to be reported. Okay. This is basically a clinical trial design which actually is. So pre-study, study, and post-study. Okay. During the so pre-study is all the preclinical trials and everything. Post-study is all the submission. You submit all the data to the FDA and everything. That is post-study. During the study which we have different segments of a study. So first is the screening visit, then is the treatment visit, then is the follow-up visit. So within the screening visit, you may have two visits. If you see, epochs are basically the visit or basically the high level segregation of a study or the phases of a clinical trial. So one phase is screening. So within one epoch, there may be more than one visit that might be there. Okay. So the first time when you visit, you are going through a screening phase. Once you take the treatment or take the drug, you are in the treatment phase. And once you have completed your treatment, and then also you are visiting the hospital to give your lab reports, blood reports, urine reports, and everything. PCG reports, then you are in the follow-up phase. So these are called epoch. And within this epoch, within this screening visit, within this screening phase, I'm sorry, there might be multiple visits which are there. Okay. So when the screening phase is going, you visit the hospital twice. When the treatment phase is going, you visit the hospital 3 times. So this is visit 1, visit 2, this is visit 3, visit 4, visit 5. So there has to be 7 visits you have to make to the hospital, each with a different agenda. So if you see for visit 1, you will be signing the informed consent only and go home as a part of screening. Visit 2, you will be filling up the inclusion exclusion form. So I'll quickly show you the inclusion exclusion form, what exactly it means, like what are the inclusion exclusion criteria. I'll show it to you. These are the inclusion criteria. Okay, if you see, healthy males, non-pregnant, lactating female, age 14 to 18 at the time of enrollment. When you are enrolling, is this one of the conditions that you are fulfilling? Inclusion is all should be yes, and exclusion criteria is all these should be no. Like history of cardiac disease or a first or a degree relative when less than 50 years of age. If this person has something like this, history of cardiac disease in a first or second degree relative when less than 50 years of age, if the person has it, and since it is under exclusion criteria, if the person says yes, that person will be out. All the exclusion criteria should be no for that participant. All the inclusion criteria should be yes for the participant. Then only that person is eligible. In all the inclusion criteria should be yes, and all the exclusion criteria should be no. Then only the participant will be eligible to take part in the study. Clear? So the next day when you come, you give the inclusion exclusion form, you sign. Then comes the first day in the visit 1, you also do one part with along with informed consent. You give your demography details, you give your vital sign details, and if you have any adverse events, you also give them. Or if you don't have any adverse events, it's fine. Then on the second visit, during the screening phase, you come and you give the inclusion exclusion form details. Okay. Then during the treatment phase in visit 3, what do? During the visit 3, if you see, you take the drug. So suppose on visit 3, visit 4, visit 5, you take the drug. Okay, and along with taking the drug, what all you do? You also give your lab results, you give your ECG results, you give your vital sign results, you give your adverse event results. So everything, all the crosses, below the visits, these are the forms which will open up in front of you, basically. So exposure is a form. So the moment you take the drug, you open the form. You take the drug, you open the form, or the nurse opens the form who is monitoring you at the site and will fill up the details. That person will also ask you if you are facing any adverse events. If you are facing, then they will include that in the form. If you are not, then that's it. If you are facing any adverse events, the adverse event will be recorded, and also a medication would be given to counter that adverse event, and that one will go into concomitant medication. So as and when we are discussing these things, look into study about these forms. I'll share with you a document, an SDTM document, where we have all these forms and all these domains listed. So what are these domains about? What are the questions that these domains have? What are those questions means? All these things are there. Okay. So this is one. So these are the 7 visits which are designed in the protocol that yes. So in the entire study that happens, there would be official 7 visits which would be there. If someone comes on any other day, suppose the scheduled visits are on SCR, day 1, day 2, day 3, follow-up 1, follow-up 2. So basically visit 1, visit 2, visit 3, visit 4, visit 5, these I have given a name, SCR, day 1, day 2 and all. So suppose a participant comes within day 2 and day 1, 102 participant comes within day 2 and day 1, or day 2 and day 3. If you see the plus sign here, So 101 participant has come exactly on the visits which have been defined in the protocol. But due to some problem or adverse events or any other thing, a second 102 participant has come on a day which is between day 2 and day 3. That will be considered as an unscheduled visit. Okay, so they also have to be kept a track. Absolutely clear on this? Any questions?
Yeah, Archit, I have one question. So, is this an example or the adverse event will be considered in the screening phase?
It's just an example. Adverse events are generally not considered in the screen.
Yeah.
Absolutely
Okay.
correct. Yes.
Okay.
But suppose, but there is a catch here. Suppose you have an informed consent signed, and after that, on the same day, you meet with an adverse event. Once the informed consent is signed, and basically inclusion exclusion also happens in visit one only after the informed consent is signed, and your inclusion exclusion is also done, and then you meet with an adverse event, then you are a part of the clinical trial now. So that adverse event will be reported. Okay, any adverse event related to the drug, not related, anything, since you are a part of the clinical trial now, anything which happens to you has to be recorded. Clear? Absolutely clear.
Okay. So, before taking the study drug, if we get any adverse event after the informed consent, it will be considered as an adverse event.
Absolutely, and you have to report that because you have signed the
Oh,
informed consent, right? And you have started the study. You are now a part of the clinical trial. You have signed the inform, you have given the inclusion exclusion, and you have been enrolled in the study. You have got an enrollment ID. That is the time the tracking begins. And after that, if you have any kind of adverse events, suppose you have got you get cough and cold, which has nothing to do with the study, that because you have not taken the study, but that also has to be reported.
okay.
Everything has to be thoroughly monitored.
So, it reported separately or with other adverse event only?
It will have some I mean, it will have a same kind of a reporting, but probably it will be later on classified as related to the drug, not related to the drug, something like that.
Okay. So, group.
Grouping will be there.
Okay. Fine. Okay. Thank you.
Okay.
Yeah.
Then this is the entire clinical trial design on a high level. Then these are the concept of a SAS programmer, which is there. Like once the data actually comes, like all the study data is locked and everything, then the clinical SAS programmer work comes in picture. Okay. So what will you have? So from beginning, what are the things that you will be having is you will be having access to the, like first there would be a raw data, because the data which you would be pulling out from the database would be raw data, because that is exactly the patient data which is there. That's why we are considering as raw data. And as a clinical SAS programmer, you will be having data access to your local system. So whenever you are joining an organization, so you will have your access to your local system, like that organization system you will have, where you will have the specifications, like you will have the documents and everything in Outlook, emails and everything you will be given. And this is also something which is not given in the local system. Everything will be there in your remote system. So if you are joining a CRO or if you are joining a sponsor company, there would be, the sponsor company will not be having a VDI, but if you are joining a CRO, and there would be a virtual desktop which would be there, which you have to log in using a username and password. And within that desktop, you will have a separate email where you will be giving, where you will have all the SDTM specification, ADAM specification, all the mock shells, the protocols, the SAS access, everything. So everything is extremely secure. And within that VDI, you will be having a SharePoint where you probably will have all the documents there. So security would be top class. You will, I mean, the organization will be having SAS licenses so that you would, I mean, by utilizing that SAS license and using your username and password, you don't have to utilize the SAS license, but it already will be taken by the organization for 50 members or 70 members, something like that. And you would be able to log into the VDI and first log into the VDI and then log into the SAS application using the username and password. Okay, and there you will probably getting the raw data files or raw files either in the VDI SharePoint, or it would be automatically connected to the SAS system. The database would be automatically connected to the SAS system. Based, I mean, as the data comes into the database, the data would be extracted through an Oracle job or a program and then would be loaded into the SAS system. Okay, if you see this relational database SQL server, proc SQL or proc DB load would be there, which would extract the database, which would extract the data, raw data from the database and load it in the SAS system, because SAS is where you would be performing the program. And now we come to the concept of standardization. Before I come to the concept of standardization, let me go through this flow chart once. So we have the raw data. Okay. Now we are coming to this part basically. All the database is locked, everything. Now we are coming to the statistical analysis. This section is being highlighted here. So first, dataset extraction, SAS dataset creation. Okay. All the raw data will be loaded in SAS and SAS dataset will be created. I think you all know about what is SAS dataset. What are datasets in SAS? I think
Yeah.
CRF is there. So based on, so CRF are all those questions which you saw. All those questions will be asked to the participant. The data will be gathered in the EDC system. The study will be closed, and all that data will be extracted using either ProcDB or user either automated procedures in place, or they would be extracted out of the database and then manually loaded using proc import into the SAS system. Okay. Once this is done, there are a couple of things. So there are three aspects to it. First is SDTM, standard data tabulation model, where you standardize the data because this is raw data. Absolutely, this is raw data. So what do? You have a ECRF annotation first created. So every questions which you see in the CRF will be mapped to a standard field based on the SDTM IG. Okay, SDTM IG would be there. So suppose if I give you an example, SDTM IG is something like this. Yeah, if you see demography domain here, okay. So if you see age, so there is an age in the CRF which we saw. In the demography, we saw an age which is there, current age. So this current age is a field which is there in the CRF. But this is not the correct one. I mean, this is the correct question, but this has to go when you are doing the standardization. This current age has to go to a field which is this one age within the demography domain. Yes or no? Because this is the standardization. Correct? Understanding? Okay. So this is one of the things. And these are all the what will I say? Formats that you have to abide by. You see, age is a number. So if you see this is the age, it should be in numbered and it is given in the CDISC notes. Age expressed in age expressed may be derived from birth date or anything, may not be available in all cases due to subject privacy concerns. It might not be available in all cases, but in our case, it is available, current age. So it will be mapped to age. You see ethnicity. Let's see if we have ethnicity here. Ethnic, we have, it will be mapped to this. So this is called annotation of CRF. So annotation is CRF is what? I will show you one more. Annotated CRF, if I show you for demography. Correct. If you see, sex is linked to sex. So this annotation is, you are annotating like the question from the CRF will be mapped to this field in the SDTM-IG. Sex is sex. You see, it is ethnicity mentioned here, but in the SDTM-IG, what is it mentioned? If you see, it is ethnic. So the field name, see, the variable name is ethnic. So it will be linked to ethnic. Okay. Region of residence. We don't have any region of residence here. What do we have? We have country. We have country. So it has been mapped as country. Okay. Visit date. When did the patient visit the first time? I mean, when did the person visit the date, or when did the person provide a volunteer written informed consent? Yes. When was the date? So that date is what? We don't have a field like this. Did the volunteer provide a written? No. That the details which are captured for this field will go into this field when you are programming the SDTM. So if you see RFIC DTC, what is RFIC DTC? Here, if you see, RFIC DTC, this is the date and time of informed consent. I will share with you this document. You can go through it and understand. Okay. So everything one after the other, one after the other, just go through it. It's very beautiful document where you will understand what is this domain related to. And you also at the bottom, you have these assumptions and dummy data as well, which you can refer to. See, we have the study ID. This is the domain. This is the subject ID. This is the start date. This is the end date, and everything is there. You will get to know about this. So coming back, this is the SDTM IG that you create. First, not SDTM IG, the SDTM data that you create. From this SDTM data, you create ADAM. This is analysis data. So first you did only the standardization. You only standardize the data. Okay. From that standardizing of the data, what do? You analyze the data. Okay. What is the average BP of the people reported who are taking the drug? What is the average pulse rate which has been measured of the people who has been taking the drug? What is the blood glucose level which have been? What is the maximum and the minimum blood glucose level which has been? I'm losing out on the word. What is the highest and the lowest blood sugar level that has been reported for the patients taking the blood and taking the drug or the placebo? So something like that. You have to analyze this, analysis data sets. So there are analysis functions in SAS. Sum function is there, count function, proc means is there, proc tabulate is there. So you use those to create the analysis data sets. And then from that ADAM data sets, you create the table listings and figures. You have the tables, you have the listings, and you have the figures. And this entire thing, you collaborate and send it to the FDA. And also there is a medical writing team who looks into this data, creates a clinical study report. Like what exactly the reports are saying, you can say kind of a short summary report of the clinical trial, and also submits it to the FDA. Any questions?
So, Archit, hello.
Hello?
I have a question related to the CRO. So,
I cannot anything that you are saying.
I guess Shabazz,
I can't hear anything
continue.
that you are saying.
Hello.
Tell me. I got you out.
Okay. I want to ask the question related CRF. So, uh, in the SDTM, like the clinical program, like clinical programmer has to annotate the CRF, right? So, it, like we have to choose the variable from the SDTM IG, or will there any drag and drop option will be there so we can do it graphically?
No, there will be, you have to choose it from the SDTM ID. So you have
Okay,
to
so we
look
have
at
to write it physically.
physically, you have to write. You have to understand the question, annotate the CRFs, get everything absolutely correctly done.
Okay.
And that for that, the understanding of the clinical trial is important, CRF questions is important, SDTMIG is important. So there is one team who annotates the CRFs, there is one team who creates the specifications, there is one team who creates the SDTMs, one team who creates the Adams and TLFs.
Okay.
Okay.
So raw CRF created by raw CRF annotated CRF created by the CDM team, right?
Sometimes it's the CDM team creates it, or sometimes it's the clinical statistical team as well. There is a part annotation CRF, like the team. I think the team who creates the specification also looks after the annotation as well. But they are a part of the clinical stats team.
Okay.
Hmm. They
Yeah.
are not a part
Thank
of
you.
the CDM team. Yes.
Yeah.
Okay, that's it. I will stop to here today. I will share the recording.